Hello everyone, welcome. In this lecture, I will try to discuss about the most important points regarding carbon dioxide transport. So carbon dioxide is a product of metabolism which can be transported in three major forms. The most important form is bicarbonate, which is about 90% of the carbon dioxide in the blood. Next two important forms are carbon dioxide bound to hemoglobin, also known as carbaminohemoglobin, and which is about 5%, and the dissolved carbon dioxide, which is also about 5%. So carbaminohemoglobin, in case of carbaminohemoglobin, the hemoglobin is bound to the N terminus of globin, and this actually favors the taut form. So as you can remember, the taut form is actually the form that's present when there is deoxygenated hemoglobin, and the taut form actually helps the unloading of oxygen because the taut form has a reduced affinity for oxygen. So here we can see the basic outline of the carbon dioxide transport. So here we have a big RBC inside the vessel. This is the vessel wall, and here we have the tissue. So the tissue, after metabolism, generates a lot of carbon dioxide. This carbon dioxide passes the blood vessel wall, comes to the plasma, and it can be carried as carbon dioxide in the plasma as a dissolved form, or can go inside the plasma, go inside the RBC, bind to hemoglobin, and can form carbaminohemoglobin, which is also 5%. And the major portion of carbon dioxide goes inside RBC, binds with water, and forms carbonic acid. This reaction is catalyzed by carbonic anhydrase, and this carbonic acid now dissociates into hydrogen ion and bicarbonate. And as the level of bicarbonate increases inside the RBC, it starts to go out. And to maintain electrical neutrality, the chloride comes inside the RBC. This is called the chloride shift. And the hydrogen ion generated in this reaction now binds to hemoglobin and forms, and hemoglobin acts as a buffer here. So here in lines you can see that carbon dioxide diffuses into the plasma and then into the RBC. Inside the RBC forms carbonic acid and ultimately bicarbonate. And bicarbonate leaves the RBC in exchange of chloride, which is called the chloride shift. And that's why the venous RBC, the RBCs inside the venous blood have a high chloride concentration. And the hydrogen that's generated in this reaction is buffered in RBC by deoxyhemoglobin. And deoxyhemoglobin is actually a better buffer than oxyhemoglobin. And in the lungs, all the reactions occurs in reverse. So bicarbonate now enters from the plasma into the RBC, and now chloride leaves the RBC. So this bicarbonate recombines with hydrogen to form carbonic acid. And this carbonic acid dissociates to form carbon dioxide and water. This carbon dioxide is expired through the lungs. And in the lungs, oxygenation of hemoglobin promotes dissociation of hydrogen from hemoglobin, because oxyhemoglobin is a worse buffer than deoxyhemoglobin. As this is an worse buffer, the hydrogen is now dissociates from hemoglobin. And this dissociation of hydrogen from hemoglobin actually helps to shifting of equilibrium of the reaction that generates carbonic acid from hydrogen ion and bicarbonate. So ultimately there is increased production of carbonic acid, which will lead to increased formation of carbon dioxide. So therefore more carbon dioxide is released from RBC. This is called Haldane effect. You can remember that Haldane effect occurs in alveoli. Haldane, H-A-L-L-L for alveoli. In peripheral tissue, hydrogen from tissue metabolism shifts oxygen hemoglobin dissociation curve to right, helping the unloading of oxygen. This is called Bohr effect. And the reason behind that is hydrogen actually decrease the affinity of hemoglobin for oxygen, and decreasing affinity shifts the curve to the right, and decreasing affinity helps the release of oxygen from hemoglobin. So ultimately helps the increase tissue perfusion by tissue, helps the increased level of oxygen in the tissues. This is called Bohr effect. So we have a practice problem here. It has been determined in healthy volunteers that the chloride content of erythrocytes is much lower in arterial blood than in venous blood. The action of which of the following is most responsible for observed difference? So you can pause here and can find the answer easily. So the answer is carbonic anhydrase. Why is so? The question is telling you the chloride content of erythrocytes in arterial blood is lower than venous blood, meaning that chloride content is higher in venous blood. So high chloride content in venous blood is the result of the chloride shift. So the chloride shift causes the bicarbonate to exit the RBC and chloride to enter the RBC, and this occurs actually due to increased production of bicarbonate due to increased production of carbonic acid, which occurs due to the activity of carbonic anhydrase. So this solves our question, and thanks for watching my video.